Okay, good morning, everyone. Welcome to the parallel session of the 20th APAMS. And of course, this is also in conjunction with the 40th and 20th and 40th anniversary of PAASE. So we're going to start first with our guest speaker. Let me check. Dr. Jerry, Professor Jerry. Okay. Yeah.
Hi.
Okay, thank you. Okay. So you may share your screen, but first I will introduce you. It's an honor, pleasure to have our guest speaker from National Taiwan University. He is currently a faculty member of the Department of Chemistry at National Taiwan University. He has two major research projects ongoing. The first one concerns the structural elucidation of protein aggregates associated with neurodegenerative diseases such as Alzheimer's disease, for which chemical methods are being developed to control the aggregation pathways of the target proteins. The second project focuses on the molecular mechanisms of biomineralization. The motivation is to understand in detail the mineralization process in organisms so that materials with novel structure and properties could be developed. A large variety of physical methods are used to study the target systems with particular emphasis on solid-state nuclear magnetic resonance spectroscopy. And I think this is the topic of his talk for today. Professor Chan had served the editorial board of the Journal of Solid-State NMR in the period of 2009 and 2016, and he was appointed as the editor of the trend section from 2009 to 2013, aiming to highlight the cutting-edge development of the field. It's my pleasure to introduce to you Professor Jerry Chan. You may start now, Professor Jerry.
Thank you, Mike. And I'm very happy to join this online symposium. So, okay, let me share the desktop. Okay. So you see this?
Yes, we can see it.
Okay. So let me adjust it a little bit. Sorry. All right. Good morning, everyone. Again, it's my great pleasure to join this online symposium, and I would like to thank Michael for his kind invitation. In the next 30 minutes, I will try to give a brief introduction to the technique of solid-state NMR, which is the bread and butter of my lab. As you can see in this periodic table, the measurements of those elements in green are, for example, lithium-7, carbon-13. These nuclei are considered to be easy or routine in solid-state NMR, and those in blue are somewhat more challenging, but still tractable. A sample in powder form is what we needed. At the molecular level, the structure could be amorphous. For example, there could be a large distribution in the bond length or bond angle, but NMR could deal with this. There's no problem because NMR has nothing to do with the diffraction phenomenon. We can also study polycrystalline materials by NMR. This is what the spectrum of a single species look like. The line shape looks a little bit strange, and it cannot be simulated by a simple function like Gaussian. You can imagine that if you have many different species in your sample, and each of them has its own signal, then the overlapping signals of this line shape will make the spectrum look very complicated. Fortunately, under the so-called magic angle spinning condition, which means we pack our powder sample in a rotor and spin it fast. With this magical technique, the signal will immediately narrow into a sharp peak, which can be simulated by a Gaussian peak. In this way, we again get a high resolution condition. Nowadays, depending on the diameter of your rotors, the spinning frequency can go up to 150 kilohertz. Of course, I'm not saying that the faster, the better. It all depends on your problems and on what NMR you want to do. As mentioned by Michael, in my lab, we have two major projects ongoing. The first one is about the study of molecular structure of amyloid fibrils. And the second one is about biomineralization, which is the focus of this talk. Biomineralization concerns about the formation of minerals in organisms. The so-called biominerals usually have complicated morphologies, but very good mechanical properties. Typical inorganic components involved calcium carbonate for marine organisms and calcium phosphate for tooth and bone. This table summarizes some of the important mineral phases of calcium phosphate. Amorphous calcium phosphate is relatively soluble. Because of its non-crystalline nature, we don't know much about its structural feature. On the other hand, HAP, hydroxyapatite, is the most stable form, or thermodynamically the most stable form of calcium phosphate. HAP and ACP are the two very important mineral phases in bone and teeth. This slide illustrates the formation mechanism of bone. It has been suggested that calcium phosphate encapsulated in matrix vesicles at the initial stage of bone formation is very important and difficult to understand. So to clarify the underlying chemistry, we want to develop a simple model system. As it turns out, we find that we can encapsulate calcium phosphate using liposomes and observe its precipitation process inside. For convenience, we refer this model system as LipoCap. Let's skip the steps about sample preparation. These slides show the TEM images for selected samples, and the time chart here indicate the incubation time of calcium phosphate inside the liposomes. When the liposomes are disrupted, or when calcium phosphate are precipitated without or in the absence of liposomes, we don't see any aggregates like that. This is a typical two-dimensional NMR spectrum under magic angle spinning measured for our sample. As you can see, there are two dimensions. The vertical one corresponds to the proton chemical shift scale, and the horizontal one corresponds to the phosphorus NMR chemical shift scale. And therefore, this technique is called heteronuclear correlation, because for each of these signals, it always has two chemical shifts. One is from the chemical shift of proton, the other one is from phosphorus. And the acronym of this technique is called HECOR. In this spectrum, you can see that there are three major components. Let's consider the top one first. With this chemical shift scale, we find that this signal has a proton chemical shift about 0.2 ppm and a chemical shift of phosphorus of about 3.2 ppm. With the spectrum of hydroxyapatite as our reference, it is very clear that in our 3-hour sample, we had already got some simple, very small hydroxyapatite crystallized. We were also able to prepare pure amorphous calcium phosphate as another standard. The signal of HAP is very broad in both dimensions because there's a big distribution of the chemical shifts. That's what we expected for amorphous materials. By comparison of these two spectra, we say that these two components probably is due to a disordered phase inside this sample, and its environment is very similar to ACP. And the signal indicated here for this one represents the phosphorus species existing as orthophosphate in close proximity to structural water. In this cartoon, we use triangle to represent the phosphate ions, and the circle represent the water molecules. For the second component, the species should be hydrogen phosphate. Here we see that this triangle is touching upon this circle as a representation. As the incubation time increases to 7 hours, we find something very interesting. The signal of this disordered phase get narrowed in the phosphorus dimension, but it remained very broad in the proton dimension. This feature suggests that the disordered phase has more ordered arrangement of the phosphate ions, but it remains an amorphous phase because of the random distribution of the structural water, and therefore we name it as amorphous matrix 2. Eventually, when the incubation time goes to 17 hours, the majority of the signal has transformed to apatite, as we have told you that hydroxyapatite is thermodynamically the most stable form of calcium phosphate. So the results make sense. So now we have two pictures. One is amorphous matrix 1, amorphous matrix 2, and these are the hydroxyapatite crystallized. But what about their relative proximity? Are they close to each other, or they are physically separated? To answer this question, we try to use the so-called spin diffusion experiments to probe their relative proximity. We don't want to go into the details. Here, I just want to show you the results. The fitting of the normalized data look very good. And the idea is the shorter or the smaller this time constant, the closer the proximity of the two phases. So from this data, we infer that the apatite phase is well embedded in the disordered phase. Overall, we conclude that the apatite domain actually grow at the expense of the amorphous domain. Instead of this, some people would say it could be dissolution with precipitation for its phase transformation. But here we find that it's not the case. It's probably occur via the solid state transformation pathway. With this simple model system, of course, we can further investigate the modulation of the crystallization kinetics in the presence of organic additives or proteins. In addition to model systems, we of course can use NMR to study biominerals such as teeth. To me, the most fascinating teeth sample are from sharks. Yes, because sharks do not have dental caries, even they never brush their teeth. Of course, the main reason is that sharks will replace their teeth continuously. But it is also important that the major inorganic phase in shark teeth is fluoroapatite. And we know that fluoroapatite, FAP, has much lower solubility than HAP. And therefore, FAP can better resist acid erosion. That's why you will most likely find some sodium fluoride in your toothpaste. But is FAP the only fluorine-containing species in shark teeth? I hope you agree that the best method to check is fluorine-19 NMR. My student somehow got a shark head from a local fish market. we took the teeth at different regions, which are presumably at different growth stage. The fluorine-19 NMR spectrum of shark teeth looks more complicated than what we expected. Here we have two synthetic compounds as our reference. One is FAP, the other one is calcium fluoride. The peak labeled by asterisks are the spinning sideband of the major peak, so we don't need to worry about this. Overall, we find that there are at least 4 different species in the spectrum of the shark teeth. You can immediately tell that the major species is FAP, as expected. However, for really pure FAP, we should not see this shoulder. So a more careful examination of its synthetic procedure reveals that this shoulder peak actually comes from the calcium fluoride. And therefore, with this, we argue that this shoulder we observed in the spectrum of shark teeth is also due to calcium fluoride. And here is the fluorine-19 and proton head core spectrum. For comparison, we also show the 1D fluorine spectrum we just shown in the previous slide. And this trace in gray is the projection of this 2D spectrum along this proton dimension. And we can see that they look somewhat different, but that's expected because of their difference in technique. Nevertheless, we just need to focus on each of these signals. And for the first one, based on its correlation peak position, we assign this as hydroxyfluoroapatite, which means that its lattice is apatite-like. But for some of the hydroxy ions, it has been replaced by fluoride ions, or the other way around. And its peak position actually appears at here. So with this, we argue that for the teeth, shark teeth, the major component is FAP, but it also has a component of HFAP. And this is the first evidence or the first such statement due to a Solid-State NMR. And the second correlation peak, as you can see from its chemical shift position of the fluorine and proton, we assign this to calcium fluoride embedded in amorphous calcium phosphate matrix. And for the third one, after checking its chemical shift and compared with the literature data, we assign this as the defective site of the apatite lattice. With this, we are able to deconvolute the fluorine NMR spectrum of the shark teeth. You may wonder, hey, Jerry, you can fit anything you like with these 3 Gaussian because the spectral feature here is not very good. But I would like to emphasize that the peak position of these Gaussians and their associated line width are actually determined by the previous head core. Here in this deconvolution, only the intensities are varied. So therefore, we are confident on the quality of this fitting. Using this technique, we can repeat the measurements on shark teeth at different growth stages. We want to see whether there are some data trend in the speciation of FAP and HFAP. Well, however, we don't see any. Anyway, we do notice that there is a significant amount of defective site in the outermost teeth. This is not surprising because the OA sample, which denotes the outermost teeth in the anterior region, this is the most mature teeth and will be very soon replaced. So this finding that it has a lot of defective sites is not surprising. Perhaps the most interesting information in this slide is that there's a significant amount of potassium fluoride present in the structure. We don't know why. It may be a very interesting issue for further investigation. So that's all for calcium phosphate. And now I would like to switch the gear to calcium carbonate. In recent years, it has been well accepted that magnesium stabilized amorphous calcium carbonate is the precursor phase of biogenic calcium carbonate. One argument is that the non-crystallinity of MgACC can be exploited to build many fascinating architecture in Mother Nature. To characterize the structure of MgACC, we again suggest solid-state NMR. The sample preparation of MgACC is relatively simple. We mix the solution of the cation and anion in a very small volume, and then collect the precipitate in ice-cooled acetone. Then using ICP mass, we find that the magnesium content of our samples really depends on the pH of the solution and the initial ratio of magnesium ions to calcium ions of the mother liquor. Next, we want to find out whether the environments of the carbonate ions has anything to do with the magnesium content. So we have carried out carbon-13 solid-state NMR study on these 5 samples. And this is the typical results. Again, we find that the line shape is asymmetric, and we use 3 Gaussians to fit it. For this component, for the ACC, amorphous calcium carbonate, the peak position and the peak width have been determined from the spectrum of pure ACC. And the same is true for amorphous, for the component of amorphous magnesium carbonate, AMC. And this one is due to bicarbonate. And with this deconvolution, we were able to determine or estimate the relative amount of ACC, AMC, and bicarbonate. The results of these 2 samples, when we look at its magnesium content, the results are in very good agreement with the ICP mass. However, for the rest of the 3, the agreement is not good. Why? Well, we suspect that here, when the magnesium content is high enough, it will aggregate to form AMC. And therefore, the signal of AMC represents nicely the total amount of magnesium in the sample. However, for these samples, the magnesium ions could well disperse in the matrix of ACC. And therefore, we don't see many signal of AMC. Is it the case? Are these ACC and AMC are well mixed in our sample? We can answer this question again by solid-state NMR. This time, we use the technique of homonuclear correlation because both dimensions correspond to carbon-13 NMR. And the way to read this is similar to what we have learned in HECO. For example, for this red trace, it's a narrow ridge. So the ACC component will correlate to ACC component. That's not interesting. That's what we expected. Again, in this position, we say the AMC component is correlated to AMC component. And now when we switch on the interaction time such that we let the ACC and AMC interact. So if they are in close proximity, they will talk to each other. Indeed, when we look at this black trace, we find that the signal of the ACC, actually, it will correlate to the signal of AMC. And this is the crosspeak between these two components. Let me repeat again. This is a crosspeak, okay? And its one origin is here due to the AMC. And another origin, when we project it into this dimension, is the ACC. And therefore, this peak is a strong evidence saying that ACC and AMC are in close proximity. So we draw this cartoon to represent what we have found. Furthermore, we can use Solid-State NMR to show that there are 4 different carbonate ions and 1 water molecule surrounding each of the magnesium ions. And therefore, with that, we could use NMR to show MgACC is not completely random. Well, I think I'm running out of time, and let me skip this. And but before I finish, I want to emphasize again that in this talk, we have just covered the NMR of proton, fluorine, phosphorus. And in fact, there are still a lot of opportunities out there. And I hope you can find your favorite element in the green squares. Finally, I would like to thank my longtime collaborator, Dr. Huang, who is the NMR manager of our NMR facility. And thank you very much for your attention.
Thank you, Professor Chan, for this very informative presentation on solid-state NMR. In fact, I just learned the interesting applications of this instrument. So I'm just, I can give you one quick question because of the interest of time. We have to proceed to the next speaker. But can you give us like a brief answer on the potential application of this to a more heterogeneous material like geopolymer? Is it possible to characterize, for example, phosphate-based geopolymer using solid-state NMR?
Yes, of course. For the geopolymer, can you see the slide again?
Yes.
So for geopolymer, I think a silicon NMR would be very useful, and aluminum NMR would be very useful. And therefore, I think we can use it to characterize the environment of geopolymer. And because, as I said, NMR can be used to study amorphous or crystalline materials.
Thank you, Professor Chen. So please stay in this webinar because after the first half, we will have a Q&A forum. Of course, there are questions from the participants. We will address in this webinar as well. So thank you, Professor Chen, and I hope you will be still here until 10:30.
Oh, sure.